Hi folks, this is Miss Dunn and we are picking up where we left off in class today. Sorry we have to go through this stuff so quickly, it's just I don't want to put us behind, and so we have a 3-day weekend for you guys, so hoping you guys can handle it. So we said before that we're not getting the 1-to-1-to-1 ratio of these offspring flies like we thought we were going to get. So according to Mendel and the laws of independent assortment, we should have gotten something like this, a 1-to-1-to-1-to-1 ratio of all the different types of flies. Why is that disappearing? But we didn't get that. We got some of them that look like the parents, actually a lot of them, and in relatively equal number here, and then a relatively equal number of recombinants. And so we want to figure out why do we see those patterns, okay? And so we, just to review here, we went back up and looked at the chromosomes, okay? And so remember, you have 2 copies of each allele, one from your mother and one from your father, okay? So all of our alleles and chromosomes come in this way, all right? So this one right here had 2 chromosomes, both with brown information for the body color and normal wings. Excuse me. This fly right here, homozygous recessive, we mated that together to get the F1 generation. And the F1 generation in all these types of crosses, this just means it's kind of a hybrid. And so it's got one big, one little. Okay, so heterozygous is basically what the F1 generation always is. All right, and so this resulting fly is going to be big B, little b, big W, little w. That's going to be its genotype. And so we're going to take that fly, okay, here's his genotype again. So we're going to take this fly and we're going to test cross it. And we always test cross with that recessive phenotype here. So these are this little guy's chromosomes right here. And so the way you want to think of this is that when it comes time, let's say this is the female, when it comes time for this female to make her eggs, she's not going to put both of these chromosomes into the egg. She's just going to put one. And as far as sperm goes, this fly is just going to put one of these chromosomes. And they're both the same. So in his sperm, there's going to be a little b and a little w passed on. He really doesn't have any other option. Okay? Now, for her eggs, this big chromosome could come in, or this recessive chromosome could come in. Okay? She's going to make either or. Okay? She's either going to make this zygote or the other zygote, okay? And you can see that if she passes on the big B and the big W, that offspring is going to have traits that look just like her, okay, with the brown body and the normal wings. If she passes on this situation, well, then the offspring is going to come out like the male, okay? They're going to look parental in that case. So that's why we see so many that look like the parents, okay? So if you come down here, you see that a great majority are parental. So how do we get then these recombinants, all right? If these are on the same chromosome, why do we get these? A little bit of shuffling here. And that we said was due to crossover, okay? And so let's think about how that crossover could happen. These are going to copy here. Remember, during S phase, identical copies are going to get made. Okay, kind of hard to fit in there. But remember, these are going to form a tetrad and crossover is going to happen. So there's a chance, and it looks like it's a fairly small chance, but there is a bit of a chance that these could switch. And so we're going to get a new formation here. We're going to get the big B and the little w together. And over here, we're going to get the little w and the big, little b and the big W. And so these could be possible alleles that could end up in our eggs. Okay, so that explains the recombinants. So these guys are a result of crossover. Okay, now there is a calculation that geneticists do, and it's called the recombination frequency. And it is the number of recombinants divided by total offspring. So it's a measure of how often crossover happens between these 2 traits on the chromosome. Okay, so the recombinants are these guys. They don't look exactly like the parents. So we're going to go 206 plus 185 divided by the total number of offspring. So we're going to add everything together here. Okay. All right, so you want to remember your order of operations here. Use your parentheses. Okay, and hopefully if you guys did this right, you got 0.17, okay? So we say that there's a 17% recombination frequency or crossover frequency. Now, the higher this number is, it indicates to scientists that the further apart these traits are on the chromosome, okay? So if you think about it, if you have a chromosome and you have all these different alleles on there, okay, and let's say over here. Okay, so basically, crossover is a random event. So it's going to happen anywhere, okay? So let's just say crossover happens right there, all right? It's pretty rare. Considering how long this chromosome is, it would be pretty rare to see the crossover right there, okay? So we would see a low recombination frequency between B and C, okay? Whatever alleles these coded for, if you did this whole experiment with those traits, you would see a low recombination frequency. Now, if I was to test this trait compared with this trait, well, the odds of the crossover happening between them would be much higher, okay? So it's possible that it could happen anywhere between those, and that's going to be quite a higher recombination frequency. So let me show you how this could be used. Okay, so we just calculated 17% recombination between the W and the B alleles. Okay, so we went through that whole experiment to figure out this crossover crossover frequency. Now, we can do this with other alleles too, okay? So this is just an example. So there's gene A, D, and F. And if we compare the crossover frequency of all of these, then we could get an idea of where these alleles lie on the chromosome. So it looks like A to F has the greatest frequency of crossover. So those, I'm going to put those very far apart here, okay, because the crossover frequency is high. Now, if we compare D to F, that's 10%, and A to D is 4%. So A and D should be very close together because it's pretty rare that the crossover happens there. So that's 4%. This is 14%. So we would think that from D to F would be 10, okay? And that is what we see because 10 plus 4 would equal our 14 here. Okay, so we can conclude that the order of the alleles here is ADF or FDA. But anyways, D is going to be in the middle and closer to A. So what I want you guys to do, this is quite a bit more difficult. We have a lot more going on, but I think you guys could do it. So use these frequencies to figure out the order of these alleles, okay? So put A, B, C, and D in order. And so start with here. A and D are the furthest apart, so kind of just put them far apart and see where you can go from here. So give that a try. I'll check that next class, okay? So you can pause the video and work on that. Okay, we're going to move on to some more kind of lab stuff that we could do with genetics. And so I want you guys to look at this example here. So we have a white-eyed female and red-eyed male, okay? So it looks like the color is X-linked and red is dominant, okay? So let's define our variables here, okay? So a regular X chromosome would be red. An affected X chromosome would be white. Okay. And so let's see here. So let's see, we got a white-eyed female, so she's going to be this, and a red-eyed male, so he must be this, right? All right, and go ahead and do the Punnett square here. Pause the video and do that Punnett square. So you guys should have gotten this here. And so let's see if the data actually matches. Yes, 56 and 58, because we would roughly expect a 1-to-1 ratio of those two, and we actually do see that pretty closely. So that's all good. Let's keep going here. So it says the F1 generation was mated together. So we're going to take these males and females and mate them together. So go ahead and take those female children and the male children and mate them together. Pause the video and do the Punnett square. Okay, so we see that we should get a little bit of everything, right? This is going to be a white female, red female, white male, red male. So we should get a 1-to-1 to 1-to-1 ratio. Do we see that? Kind of. This 62 is a little bit troubling, perhaps. Overall, it looks pretty good. The question is, do we have the mode of inheritance right? Is this actually X-linked? Maybe we're looking at this wrong. Maybe it's not X-linked and it's just it's on a normal chromosome or something like that. So what we can do whenever scientists think they have it figured out what the mode of inheritance is, they always want to do a chi-squared test just to be sure that this data is close enough to our expected data, okay? So we expect that we would get equal numbers of all of these. This is our actual numbers. These are our observed numbers, okay? Okay, so count up the total number of individuals, divide by 4. That's going to be our expected number, okay? And then we're going to plug into the chi-squared equation, and you guys are going to figure out all that stuff. So calculate the chi-squared number and try to fill this all in, okay? And then you have some practice problems to do. And so you're going to go ahead and do all of this. So the table here, remember, always look at the 0.05 probability. So check out your degrees of freedom. Since there was 4 boxes of our Punnett square, remember you want to take away 1 from that and look here, okay? And that's for the example that I showed you above. This one might be a little bit different. Okay, so go ahead and complete that to the best of your abilities. We'll be going over that next class, and I'll check that off. So make sure you try everything. Don't leave anything blank. Give it the best shot you can. Okay, see you in class.